I work with José Onushi Group in the Center of Theoretical Biological Physics. And I'd like to start thanking Nathan and the committee to give me opportunity to show some of my results that I've been working the last 3 years in this new area that is genome organization. So my seminar today have the title Physical Modeling of the Genome, Structure Insights and Biological Consequences. So here I have an outline that we're planning to talk today, is try to explain the genome organization problem to try to motivate you to how you can apply some physics modeling to understand this complex about the genome organization. And then I will talk a little bit about the high-C experiments. That is the most modern way to extract information of the contact maps of the genome organization. How you can modeling that. I will talk a little bit about this modeling that called Micron. That's the first model of our group that have very successful to explore some details of the chromosome. My first contribution for this system, that is the OpenMicron. That's a software that can make chromosome simulations more fast, and how you can apply that. And to finish, I will show this new idea that I'm been working the last year, and probably will be the next generation of modeling for chromosomes. So to start, let's understand a little bit what's the genome organization problem. If you get just one cell for your body, the number of base pairs that compose your DNA is an order of 6 billion base pairs. If you make estimation that each base pair, they have a 0.34 nanometers of length, and you stretch out all your chromosome, you take along almost 2 meters of DNA, right? So the idea is, okay, these 2 meters of DNA need to be packing in a cell nucleus of 10 micrometers. So this is the first challenge. I think this represent good the idea because you have a lot of information, a lot of DNA that need to be packed in a very small volume that is the nucleus of the cell, right? This is the main challenge that you have here. And not just packing, because we need to remember that inside this nucleus, you need to make the replication. That is when you divide the cell, you need to expose some parts of this genome to express genes. So this be very well organized. It has happened like in January 1 day, 24 hours, you have these replications happen. So this is the big challenge that we try to face and understand. So for the point of view of the some experiments, you have this idea that the first way to packing the double helix here is when you wrap around this proteins that called histones. Here's the crystal. The first time that they did that is in 97. You have this crystal where you can see the DNA is wrapping around these proteins, right? You have some images here that show, for example, that in this part here, that you can see this nucleosome. That is the combination of 4 of these histones wrapped by in the DNA. So you can see some images that what has happened. And this nucleosome started to make like a helix that they have a name of tight helical fibers here. You can see that you have some images that they can visualize that. So what's happened is after this, for this point, until the chromosome, like for example, this is an example of the chromosome metaphase, you not know how it's organized. It's very hard to make imaging. These images is just starting to bubbling this kind of experiment right now. So it's still a challenge to understand what's happened from this part to another part here. More recent images, you can see, for example, that Kramer in 2005, they showed that if you look the chromosome nucleus, I'm sorry, the genome, the nucleus of a cell, you can see that each chromosome, they remain keeping your own territory. They're not entangled together. So in general, you can see that each chromosome have like this all position. They are not mixing. Another information that you have here is like for the human chromosome, you have 22 autosomes. They are pairs, so you have 44 plus 2 sexual ones. So in total, you have 46 chromosomes in the human cell. Okay. Okay. So how we can understand like this?
You said that they don't overlap, but what was the alternative? Could they have overlapped?
No, they don't entangle. This chromosome in general, they just keep together, but remain this each chromosome in our own territory.
But could it have been different? How could it have been different? They occupy some space, no?
Yeah, this is what the experiment showed to us. What can happen is that they just entangle all together. That's something that is a little bit weird. The experiment showed for us that, okay, each chromosome, make a wrong comment here. It's not supposed that, for example, this chromosome 6 here, they not remain here all the time, but they, in general, they not entangle. So they have movement around the nucleus, but always using this phase separation between these two chromosomes, or all the chromosomes. Is it correct? So this experiment should just show for us that all the chromosomes, they not entangle between them. They don't make like a mix of like a pasta. They remain in separate regions, like territories. This is the experiment showed to us.
Okay, I get it, yeah. So it's, I see, okay.
Okay, more questions?
Yeah, can you just say what can we see? What is just the scales, just so I understand the scales? So what is actually possible to see now in an experiment? At what scale?
So when I look for these images here, you just, okay, you can understand because you can create this crystal and understand like this first packing here, right? There's another images here show this region that is around 30 nanometers of diameter, these fibers. But after here, the people don't understand well for now. It's very hard to make the imaging of this kind of packing here. We're talking about in terms of scale is 700 nanometers here. This is a very challenging experiments. It's very challenging to perform experiments in this region.
Sorry, we can see 30, but we can't see 700?
Yes.
Why?
Because in this small part here, okay, this first part, we can crystallize this region and make the X-ray, for example, to visualize.
Okay.
Until this region that is very sparse here. When you go for this region that is more compact, it's very hard to create like these images. The only way that I show here is like a very distant image, like where you see the whole nucleus here. Okay. What they are doing now, there are some experiments very modern that they released like 2 years ago. They call Sprite, that they can make some markers in some regions here in the chromosome and start to visualize parts of the chromosome in that stage here when they are all collapsed. But it's like a very new experiment that people are just starting to understand. I don't have images about that, but I can show you later. Okay, more questions? Okay, so a new strategy to understand what's happening in this part here is this new technique that is developed that called Hi-C. Hi-C is like a contact map for chromosomes that you can make some experiment that you measure the contact looking for the proximity in different DNAs. I get this image to explain this, how it works, this experiment, because it's very nice. So the idea is, if you want to make the contact map for proximity, we can, like, for example, look for the Homer Simpson Facebook, take the pictures, and calculate the frequency of contact between them and their relatives, right? There are some pictures they are alone. There are some pictures they are with his wife, his kids, some friends. So you can get all these information and create like a contact map of proximity. So this is an example that's what's happened. So looking at all the pictures, you can measure the frequency of contact of the people. And here you can see that we have this cluster here, that if you take a look, is the family right? They are more proximity for these two, the two kids here. I think this one is too young, they don't care too much about him, but this is a good way to understand that how you can create that. You can do that with the chromosome in a way that you get all your genome. Yes, you get, okay, it works. You can get all the chromosomes here and calculate the contact of each region of this chromosome are making contact with another part. So here you have the interaction between intrachromosomes, right? And the another one here is the interchromosomes, is the interaction between different chromosomes. Depends of the experiment, right? The first experiment that I will show after that, like for example this one, they have a resolution of 250 kilobase pairs. So each bean of my map represents 250 kilobase pairs, right? Just in this example.
But they're all the same? Like you just take 250 and divide?
Yes, like beans.
Because
yes.
I remember I saw different beans.
Yeah. I will show the next slide here. Just for curiosity, the experiments are made in this way. So they make a cross-link in the DNA, and you have like some cut using some enzymes. And okay, it's a whole pipeline that I don't enter in details here now, but what's happened in the end, you have a lot of these fragments that you can measure, like count the number of contacts of this specific region. And here is where you define the resolution of your map. If you can improve these small pieces here, probably your map you have more resolution. So nowadays, just to advance that, you have maps when they look like some 100 base pairs of resolution. That is very well understand.
Sorry, I'm a bit confused. In the previous, let's say, matrix or...
This one?
I mean, yeah, here you are talking about a single chromosome and the various regions of the chromosome, how do they pack, or chromosome with another chromosome?
No, here I'm just looking for the chromosome 8.
Okay.
Right? And like if you get the beginning of the chromosome, here's the end of the chromosome.
Okay.
And like this region in head, explain to me the frequency of contact of this region are made over this region.
Okay.
Over a million of cells. So this, in general,
So essentially, this is a way to reconstruct how a single chromosome packs in order
itself,
to be compressed. Okay.
Perfect. You can look, for example, if I go back a little bit here, you have this another region here that is the contact between different chromosomes. You can see for...
I mean, in the other, when you talk about a single chromosome with itself, I understand more. Here, I would think that let's say the contact between one chromosome and the other can change in time.
Yes. Like, if you take a look on this map, this is a frequency map. So if you look the intermaps, that is the map between chromosomes, you see that it's more white here. They are not frequent.
I
right?
see.
When you compare the intramaps, that's the interaction between the same chromosome.
I understand now. Each square, it's a chromosome.
Each square, yes, this red square is the intrachromosome interaction, and
I
here
see.
is the interaction with this chromosome with all others.
Okay.
Okay? I'm sorry for not explaining well with that, but like
Sorry,
here,
no,
when you
I'm
look at
confused.
chromosome 8,
Chromosome 8 is one chromosome, right?
mm-hmm.
You're mapping the interaction of that chromosome with other chromosomes?
Yes, the interaction between them.
Other chromosomes.
Yes.
But you started by saying that the chromosomes have different territories, so why do you care?
Yeah, but they are neighbor, right? They not entangle itself, but they are like general neighbor.
Sorry, what problem are you trying to solve? I thought you were trying to solve how the chromosome sits by itself. You're trying to solve the whole problem at once with all the chromosomes?
What try to understand here is how they are organized.
All the chromosomes.
All the nucleus, yes.
That sounds
But
like an incredibly hard problem.
yeah, but like we start looking for a single chromosome, and then you can explore
No,
how the
so I
interaction between
understand
them.
starting with a single chromosome, but then why do you care about chromosomes interaction with other chromosomes if you're starting with a single chromosome?
Yeah, you. I think in the next slide will be more
Okay,
clear
okay.
what we can, what's defined that you, right? Because I can take a jump on it that say that there are some regions on the nucleus that they expose some regions for different chromosome for the same area. That's like, you can understand like a machinery that, for example, express genes. So this is a region that they call speckles, that you can put some regions for different chromosomes in the same area that can improve the production of genes, for example. So what's happened, they organize in the same way that they put all these regions that are supposed to be expressed in the same region to be expressed. And there's another part of the chromosome that you call heterochromatin, that in general not express anything, it's just information that need to be replicated for different stages of life. In general, they are more compact and stay more on the side. Okay? Mm-hmm. So the contents here, if I go back in the experiment, sorry, it's forward.
Is there a measure of a distance?
Yes. But in that case, they are measured by this kind of, in the experimental part, we're not talking about the simulations or the modeling itself for now. But the contact is made by when they make these cross links. You have this protein that can glue here, and this can't for one contact. They are very close in proximity, right? This is the experimental way to do that. Okay? So if you look this first paper in 2009, the Aries Aiden, that's the guy that create together with this Job Decker. You have this map, for example, this one is 100 kilobase resolution. What's happened is each of pixel of this map represents 100 kilobase pairs, right? And then there's the contact map. So I just explained that, I will pass that, but the idea is that, okay, you have some region J and region I, and can measure the frequency of contact between these two regions here. Yeah, I already explained that. But in 2014, Aries give this map that have the kilobase resolution map. And here start to appear some features that we start to take attention. This intrachromosome is just a chromosome, sorry, a little bit blind, this chromosome 19. So you can see that have this check border pattern here. There are regions that is more likely to make contacts. There are another region that don't like to make contacts, right? They define these chromatin types. So you have type A's and type B's.
Sorry, why the diagonal is not enhanced like in the other ones?
Because it's, if you look the chromosome here, I'm just zoom in this region here. So it's like this. It's like I'm giving a
you're
zoom
off
in this
diagonal.
region.
You're off diagonal. Okay.
Yes, it's not the main diagonal. Perfect. Just to explore that we have this region that have this checkerboard pattern here.
And just out of curiosity, so
Yes.
this is a frequency map. So how many times?
For this
have,
over this million of cells that I make the experiment,
millions
whole
of cells.
time they are in contact or not.
Okay, so millions.
Yes, this is the order of
Yeah.
millions. In that case, when you go to in higher resolution like this map here, you need much more information. Even because like the inter maps is more hard to find contacts, so they need to explore these different kinds of contacts more often.
So to make this experiment, to get this data, how long does it take? Just out of curiosity.
Oh, this one is a big one. Now they change this experiment they call in situ high-C. Now they change for a modern pipeline that call intact high-C. That is like, in terms of 1 week, you can have this map if you have the pipeline doing. And a good thing about that.
How long? Sorry.
Like 1 week.
1 week?
Yeah,
That's
all the
not-
process.
That's not bad.
And the most interesting part, this is a kind of experiment that's cheap. You not need like very expensive machinery to do that. This is one idea that I wanted to bring this kind, because don't have any groups here in Brazil that make this kind of experiments. They're cheap. Well, I can talk about later about that, but this is like a new experiment that is cheap and very interesting to us to understand that, because there is a lot of, for example, you have a collaborator that they create a website that called DNA zoo, that they are exploring around the world a lot of different species to try to mapping and see the difference between them. And Brazil don't make part of that because it's a little bit of bureaucracy to make this bridge. So this is one point that I'm interested in the future to make this bridge between these people. That's our collaborator for some time, and bring that because you have a lot of species just happen here in Brazil, so maybe it's interesting. But I will show in the results some of these ideas. Just to continue here, you have these types, A's and B's, where this B types is more refers more about the heterochromatin, this part of the chromosome that not express too much genes. So you can see here that this region in general, they are more collapsed. So you can see these big red regions here. And the chromatin type A is more related to regions where the genome are expressing genes. So you can see that. And there's another kind of experiment that's the epigenetics experiment, like enrichment of some proteins, for example, some methylation stones, that you can see that correlates very well. If you get a B part here, you can see, for example, these experiments correlate very well with this region here. And another feature for the experiments is like, if you get the same DNA for, like, you get your body, get a sample for your blood, and a sample for some specific tissue, what you can see that for the same region, the contacts that is made for these different cell lines is different. So here you have the same DNA in different part of the body that they are like they are organization in different way. So you can see that change depends of the part of the body that you are studying, right? Exactly, like for example, a tissue that's more interesting to express genes that's related to more like something of the tissue, and the blood is another part. What's happening is the chromosome, the genome, they organize in the same way that expose some region that is more important for this kind of expressing. Okay, just all the features from Hi-C. I just talk a little bit about the compartments. When I say this about this 1D tracks, this epigenome information, you can split this A and B compartmentalization in subtypes. For example, A1 and A2 for type A, and B, we have B1, B2, and B3. What's changed between that is that some of them is more related to some of these epigenetic marks. So this is a way that you, when you look for a genome, you can get the sequence annotation. You can understand what part of your chromosome is type A or type B. This is something that is very easy to do. Type A and type B, you just make the Pearson correlation and get, for example, the first component of principal component analysis, you get the A's and B's. Like for the subtypes, you need to make more hierarchical clustering of these Hi-C maps to get this information. Okay? Okay, so now we're just talking about a 2D maps, right? It's just a contact map. It's like a 2D information. I know that different parts are made in contact, but what's happened in the 3D? So how I can look for this 2D map and understand how it's occurs in 3D structures, right? In this way, that we'll start to make the chromosome modeling of that. The question is exactly that. How I go for the 2D map and go to the 3D example of a structure. So what we're using here is like this top-down approach because we want to extract information for the experiment to create your modeling. You're not going for like first principles to understand that because we need to understand more general details before. So this is a choice. To do that, we use like classical polymer physics. That's you can get like a bead, connect with springs, right? And then in the top of that, you add some features that you can extract from the Hi-C. In that case, you use maximum entropy approach to do that. In other words, you have like, you want to make like Langevin dynamics with that, so you need a energy potential to work. Here, I have this homopolymer potential that comes from the polymer physics, and I have the second potential that we call observables that can change depending on the way that you are modeling. In that case, we are using this idea where each of these lambda is like a Lagrange multipliers that you need to training, you need to optimize to find what the best value of that to put here. I went in details about how looks like these potentials in the next slide. But the idea is, after you have like this Lagrange multipliers optimized, you can run this Langevin dynamics in this case for generate an ensemble of structure, and then with these 3D structures, you make like a high C map for that, like we call in silico high C. You measure the distance between the chromosomes for a single chromosome in this case, and then you can create a contact map that is coming from just this ensemble of 3D structure. In a way to measure if it's okay, you can compare to the experimental maps here and say, okay, if this map is good or looks like they have the same features, maybe you can believe that your 3D structures, they have some important information about the dynamic of this genome. Okay? Yes. To make this minimization is a little bit hard because you have like multiples of these Lagrange multipliers. You have to do, in some parts, the second-order optimization to find the best value for that. And you become a little bit problematic. If you work with some machine learning, you see that when you need to make the inversion of some Haitian matrices become very costly computationally speaking. So sometimes you need to avoid that. But
Sorry. Since you see, let's say, discrepancies between the experiment and the result of all this machinery, do you think the discrepancy is because, let's say, the function that you are trying to minimize is not the actual function of the chromosome, or you simply got stuck in a local minimum for these lambdas and there will be a better global minimum? Which of the two?
The two possibilities exist. You need to take a lot of attention in the training of these potentials because you can be trapped in this local minimum.
Right.
Is it correct? And the second one is like, depending the way that you define these observables, is not enough.
Yeah. I mean, I was wondering if it is, let's say, how you design this function. I mean,
yes. Here's I will show to you.
Okay.
We have different models. The first one that is very successful in our group is this minimal chromatin model. That's the idea. Okay, let's put minimal physical principles on that. I can show that you split. So you have the homopolymer potential that you can discuss after. This more simple, you're not training that. But here, these observables that I showed to you, you will split in 3 different functions here, right? That we believe that is the principle for that. The first one, for example, is the type interactions. Remember the types, the subtypes that I explained for the experiments? So you have like a potential that measure how the interaction between different parts here. So this model is like a copolymer model. You have like flavors for my beads. And these flavors can repeat. You can see that I have like just a schematic way, but you have like A's, A1, I'm sorry, B1, A1, all this stuff. So you need to train in how the interaction between each different flavor is this is the Lagrange multiplier for that case, right? You need to train that. You have the second part that is the loop formation. This is a new feature that when you zoom in some region very close to the main diagonal, there appears this feature that the DNA likes to make small loops. So you capture this information from the IC2 and train that. This is a little bit more simple. It's just connect or not connect here. And the last one, you call it ideal chromosome. This is the Lagrange multiplier that we need to train in. That's, if you take a look in this potential here, they just refers to how distance in genomic. How is the genomic distance between these 2 bits? This measure more the how compact is your chromosome.
But the functional form of F is derived from where? I mean, here you put F, R, A, J. What is the functional form of this F and how you determine it?
Yeah, this is like a choice. This FR for us is how you measure the content itself. If I have this in my last slide. Where is my?
No, okay. If you're gonna comment later,
Yeah,
we can wait.
like in the end, I
Okay.
can show to you. But
So
what,
just to anticipate, is something that, let's say, is inspired by the experiment. Like the experiment give you insights on what is the correct
The
F.
part that you infer for the experiments is training these parameters. This F of FRIJ is just like how I measure a contact in a polymer way, right? I have a distance between them, so I can measure that just have a contact or not, like a binary way, or I can have like a tangent. In this case, you use a hyperbolic tangent to define like how much is this contact here. I think it's good
Okay.
to show to you how you can calculate that. Just let me go back. Yes, so. Okay, I just pass very fast. This is the homopolymer potential. So you have like the harmonic one that you choose the Fenn bond that have this shape. You have this angle. Sorry?
Who gave you these functions? Where do they come from?
Where it comes from? This.
Yep.
This is like a very classical way to use to simulate polymers. What they use here is like this is like a harmonic bond that measure their bonds between them. This is a Grossberg potential that they released in the paper like for some time. The people in general prefer that than the harmonic one, because you can see that it a little bit change the distance of the minima here a little bit for more close, so you allow a little bit of contact between the two bits, right? In that case here, you are included the hardcore. Of course, you don't want to do your beads come together.
Yeah, but just for example, why is there sigma to the 12, sigma to the 6th? Who gave you these numbers? Where do these come from?
Like this one is like a Lennard-Jones potential, right?
I don't know what that means, sorry.
Oh, I'm sorry. So, okay, you have this kind of Lennard-Jones potential that they have this hard wall. So here, the two beads that you are simulating, they not allow, they cross together. It's like a spherical wall, right? But when you go a little bit far from that, you have a minima.
But just for example, why is it just looks bizarre to somebody who doesn't work in the field. Why is it sigma to the 12th and sigma to the 6th? What's special about those coefficients? Or?
Yes, like this potential, when you go to the, because this is a mix of Lennard-Jones with the Fenn bond, but if you look just the Lennard-Jones, they have this kind of way. This is coming from when the people try to understand like van der Waals interactions when you have particles, where they are go very close, they need to be like repulsive a lot, but when you go a little bit far from that, you have a part of the interaction. So they attract in a way in the power of 6
I
here.
understand, but does this come from solving some equation, some kind
Yes.
of... So what equation does it solve?
Good question. I don't remember on the top of my head now, but I can bring to it
Okay,
tomorrow.
so and the equation...
Yes. Exactly. Thank you. Yes. So there's like a whole field that just try to make this analysis of how small molecules interact. Like van der Waals interactions, for example, there's something that they derived from this kind of ideas here.
And sorry, I'm sure I'm having a trivial misunderstanding. Can you go back probably a couple of slides?
Come back in.
Back, I think, yeah. Another.
No,
more.
The equations, I start here.
No, I know, but now I got confused about something that was before. Sorry, because
you just to remember, you asked me about the F of
no,
R.
yeah, that I know.
Yeah.
But then I got confused
to,
where the experiment entered, because the alpha are minimized by minimizing this function.
No, the FR is like, for example, if I go back in the potential, this is my F of R.
Right.
Right? Remember the equation that I showed there?
Yes.
You have this parameter that's trained by the experimental maps, and they have this
Ecco, no, okay, that is the question. I thought that gamma D was simply, let's say, obtained by minimizing this function, right?
Yes.
No?
Minimizing, no, we're minimizing, these parameters are obtained for the high C map. So when you make the comparison between what's happening in the experiments, we need to make like an optimizing for find what's the best values for these lambdas or other
Okay,
ones
okay,
to
okay.
fit the experimental results.
Okay, okay, thanks.
Okay? So, like you said, like the way that you define if there is a contact or not, you use like a hyperbolic tangent this way, where this mu and RC we can extract from the high C. What I can see for that, if you get like the high C map here, you need to see how is the first neighbor here. You need to check if the first neighbor is equals to 1 or not. So you need to find, you tuning these values to define what is the contact. In other words, this is a definition of how you make a contact or not in your polymer model. Okay? No, depends of the system that you are using.
No, but F appears 3 or 4 places. In
Yeah,
there.
yes. This
Use
is just
the
a
same
definition
mu?
of contact you're using all of them. What's
Mu-
happen if you change the system that you are looking, for example, you go for a human chromosome and go for like another animal, you need to not retraining, but recalculate this mu and the RC. That is very dependent of this first neighbor and the second neighbor of the high C.
So, but is it clear? Maybe it's clear to you, but not to me. Why is it the same mu that appears in all the different terms in your U?
Because this FRJ
some
is
local
just a
property
definition
the
of
chromosome?
contact.
So it's a local property of the chromosome that determines mu and RC, is that right?
Yes, the mu and RC is defined by the species, right? So I can, what happened? I can look.
It doesn't depend, for example, on the type. You had various types, right? You had
Yes.
type K, type L.
No,
blah,
no,
blah,
because
blah.
the types, when you go to the types, you need to train in this Lagrange
No, I'm not
multipliers
asking
here.
about training. I'm asking, suppose we have 2 different types of pairs. I don't know what, K and L are different types of genes or pairs or something like
Yes,
that?
can be different types. For example, A and B.
Yeah, so why doesn't mu depend on the different types?
No, they don't different. The mu that comes from this FRJ do not depend of the types.
I'm asking physically, why doesn't it depend? You say it depends on the species. Why can't it depend on the...
No, because we need to train in these parameters from
I'm
the
not
high
asking
C,
about
right?
training. I'm asking about the starting point. I'm asking about the starting point. You're assuming that all the Fs have the same mu, and I'm asking why do you assume that?
Okay, I think we assume that because it's like a definition of contact, right?
So it's your definition.
Yes, you need to
Okay.
define what is a
But it
contact.
doesn't seem to me to be a natural. If you know that mu changes depending on the species, why don't you believe that mu changes depending on the type?
I think this change that you are talking about, they will appear when you recalculate these Lagrange multipliers.
No, that's a different parameter. That doesn't appear in F. How can alpha have anything to do with mu?
Okay, let me try to explain for you. Okay, you have your polymer here, right?
Yes?
Maybe even more relevant, RC, right? RC is the proximity when you start detecting that they are close. Indeed, different species start killing each other earlier than others, right?
Yes.
Why would RC be the same for all types, for example?
I really cannot I not understand the question because like maybe I'm not explaining well. Like I have this kind of function, right? The RC is the definition where they have probability of find this contact in 50%, correct? So for example, I have A and B here is a contact. Here is my I and J, right? And this region here that's defined by the same RC is just the definition, okay, this is the defined, it's like a contact, correct? If I change it, instead of looking at contact between A and B, now I'm looking for A and A, my definition of contact must be the same. Because it's just geometric definition, right? I'm not
No,
changing.
Rij is the geometric, not RC.
It would be the same for all species. It's not like when you have particles that interact with some correlation length, right? Correlation length changes. So not all short-range interactions are the same. You have various masses, various correlation lengths. A priori, A with A could detect it themselves very easily, but A with B, maybe they need to be really, really close to know that they are nearby. Why would it be the same? Maybe you don't want to have too many parameters, and that's one reason. You just say, I want to have 5 free parameters, so I'm not going to start putting A, B, C. It's a more practical answer. But from a physics point of view, it's not very clear yet. Maybe a related question is you have your potential is super general, right? You have many, many terms.
Yes.
For example, you could start putting couplings, cubic couplings that depend on 3 terms.
Yes.
Or terms that depend on the curvature of the polymer, right? If it's very curved or not. What is the principle that tells you which terms to put and which terms not to put? So when we have effective field theory, for example, we have this type of rules. We know how to organize the terms so that some are more important than others. How do you do here? How do you know that you are not missing terms that are important, or how are you confident that your potential as the most important terms?
I think that at least for our kind of modeling is just a choice, right? You have some, you have a homopolymer potential that takes account of this like bond, angles, hard sphere for not being counted. And now the new features that you want to include in this kind of modeling, you want to extract from the high C, extract from the experiment.
Right, but connected, I think if you want to follow this idea, like I start with, let's say, more or less handmade potential, it becomes crucial to understand if the discrepancies are due to the fact that you are stuck in a local minimum or not. Because if you find discrepancies because you are stuck in a local minimum, you can say, okay, maybe the function will be good. I just stuck in the wrong place, and maybe I can do a better algorithm in order to arrive to the global minimum. If instead you are in the global minimum and still there are discrepancies, that means that your function is not good. Yes. So do you have a way to distinguish if the discrepancies are due to a local minimum versus a global minimum or not?
At least when you train that, we can look for our results and compare if you are matching good with the experiments or not.
Fine, but you don't know the reason for the discrepancy. You don't know if it is due that your minimization is stuck in the wrong place, or if it is due because your function is not expressing what you have to express. You don't know that.
But depends what you mean about trapped in the local minima.
What I mean is that you have a function.
You are making a wrong modeling, right?
No, I'm saying that. I guess you are following some gradient, right?
Yes.
Why would you care about going to the global minimum? Physics will not go to the global minimum, right? It's a very complicated potential.
Yeah, but
you can.
No, but we need to remember that when you work with the maximum entropy, I'm sorry.
To me, it doesn't seem natural to go to a global minima. I don't understand. Would you want to go to a global minima?
Not necessarily. You just wanted to find a good solution that satisfies. Yeah. Yeah. In that case, when you work with this maximum entropy approach, it's very hard to define if you are in the global minima or not. You need to find a solution, and let's see if the solution is good enough to accommodate what we expect for our result. This is a very traditional way to look for maximum entropy.
But I was thinking that biologically the chromosome arrive at the global minimum, but you say probably not. Okay, fine.
A very simple question. What are the units of R that you use in practice?
Oh, in that case, when you make this modeling, you use reduced units. So in general, our distance is measured by the minimal. When you make reduced units, you not have physical units like meters, kilometers, or centimeters, right? You definition, you start the reduced units using units of KT, that is our energy is equal to 1. And then you transform all your units in function of this parameter of the KT, right? So in general, we not have like a unit of distance. This kind of modeling, we will not like to solve exactly looking for real distance. It's just like a more coarse-grained model that we spare that find this.
So can you use like the number of bases that you have? Is that a distance or not?
Sorry?
The number of bases of, have 10 bases.
Okay.
Can you use that as a distance or?
You can use to estimate, after when I show my results, I can tell you that when you have this solution, you can estimate what's happened if you made like this selection. For example, we have like this estimation of the size of each base pair or the size of my bid in this case. For example, in my modeling, I use 50K bids resolution per bid. So each bid have 50K base pairs. So with this number, we can make some estimation of the real world values.
Okay.
Okay?
So another question, maybe you get there, but after you train everything, can you make predictions that are compared with experiments?
Sorry, can you repeat?
So after you have your model and you fit the parameters using the measurements of your experiment, can you make predictions?
Yes.
Okay.
This is part of the results that I'm planning to show. Okay, I go back to okay, just going back, I'll go a little bit more fast in this part, but the idea that after you train this parameter and make like a simulation with long replicas of the same simulation, you can compare the high C, for example. This in the upper diagonal here is the map created by our simulation, and this is the experimental map, right? So here's just a zoom in some region that you can see that these checkboard patterns you can recover very well. So these results here are trained in one specific chromosome, in that case chromosome 10, and then you apply for all other chromosomes in the human, and the results are good like this one. So I not show this result because it's a little bit old. I will show more recent results. But what we can say is that micron simulations is transferable. So you can train in one chromosome and apply in other chromosome for the same species, and you have good results in that case in modeling. When I say good results, is when I compare like this kind of high C, scaling, a lot of another parameters of the polymers. Okay? So this is my first contribution for this kind of modeling, because what's happened until this paper in 2016 is you need to borrow some tools from protein molecular dynamics for proteins, for example. They don't have like a way to make these simulations just for chromosomes, right? So all the time you need to make some change or want to include a new potential or something. It's become very hard because you need to use a tool that is not developed for that. When I come here the first time, 4 years ago, when Jose is in the sabbatical, we start this idea that we, together with Vinicius, that, okay, let's make software that you can simulate chromosomes, training potentials and simulate all this stuff. So this is what we did. We used this idea of this micro potential together with this new technology that is OpenMM, that you can make molecular dynamic simulations in GPU that's become more fast, more easy to calculate. And then you start to create this app, this software that you can make like the simulations very fast. You can show here that if you increase the size of your simulation, In GPU, you can make it very fast when you compare to CPU. For example, you can run multiple chromosomes. That's something that's very hard to do before. For example, all the results that is created by the Micron in this 2016 paper take like 2 or 3 months to run. With this new software, you can do it like in 1 day, even the training and the simulations. So when you create that, this open a lot of possibilities to explore different species, different kind of simulations, different kind of potential and training. This is the one that I want to show.
Sorry, just a curiosity. What was that graph with the wheat and sugarcane? What?
This one?
Yeah.
Here I measure like if I increase the length of my chromosome,
Yes.
right? To run the same amount of time in computational
So just to
time.
understand, the length of a chromosome of sugarcane is longer than human? Is that the statement?
Yes.
Right? Ah, I see. Okay.
Yeah,
Interesting.
like the wheat is the biggest chromosome. If you wanted to simulate the full nucleus of a wheat, it's like gigant. And it's
So
enormous.
there are many more genes in a wheat than there are in human?
Yes, there are more base pairs. Not specifically because gene is a sequence of base pairs, right?
Right.
But there are more informations in terms of base pairs, for sure.
But not more genes, or is that not known? Or
I don't know to see if there are more genes or not, but there are more information held in the wheat genome.
is there a biological explanation of that?
I don't know. I don't know exactly
anybody know?
why they are bigger, but yes, you
it have
can
to do
see
with
that the human is
mutations or
not the
something?
at least not the bigger one.
Does it have to do with mutations? Does anybody know? You guys know or no? Bizarre. So there are more chromosomes in humans or more chromosomes in...
Yes, sometimes there are more chromosomes. There are more species. They have more than 46 chromosomes, for sure.
I see. Interesting.
Like for example, I will show a result from onager, that's a donkey that have 52 chromosomes,
I see. Okay, thanks.
right? The yeast genome, for example, they have 32. It's a little bit small.
Okay.
So it is not there is not relation is the length of the genome with the
What we think is complexity. Yes.
yeah.
Okay. Okay, thanks. Thanks.
Okay. So what I'm showing here, that if you increase the size of your simulation, is more simulation stuff, it's become, of course, if in terms of molecular dynamics, become more harder to calculate. So before that, is made a little bit more impossible to calculate bigger chromosomes. Or you take like half a year to make the simulations, and now you can do it that much more faster. Okay? So that, like I said, we will open a new box to explore different aspects of different genomes. So here I have some works that have been collaboration, work together. So I will just highlight some of them. One of them is, of course, if you have now a package that can make more simulations, why not make the full nucleus simulations? So this is what I'm doing here. You have 46 chromosomes for the human genome, and then you make these simulations together with these PhD students, Mateus, that for some of the results, you can see that the maps is very high correlated. If you look, the Pearson correlation between the intramaps, they met like almost 0.95% in average, right? So now they are like looking for this kind of simulation to see how these chromosomes move around this full nucleus. So if you're, they go from one part for another. What's another question that you are asking now is like, what's happening with these big regions of type A, for example, that they like to stay together to make more exploration in gene expression, all this stuff. But this is just kind of work that you can do. Mm-hmm. Okay, I'm sorry, I just pressed that. Yes.
So you are, probably I'm not understanding. So you managed to separate the various chromosomes, as in the picture on the left,
Yeah, this is like
just by imposing rules inside the chromosome about how different types interact with each other.
Yes.
And that automatically implied that the various chromosomes also separate without putting that as input?
No, the only input that we use here is the same equation from Micron that I showed before. So I just have types interactions. This is the only interaction that interact between different chromosomes. All the other potentials that I showed there is just inter-chromosome interactions, like the compaction, the loops. The only interaction that's happened between chromosomes here is the type interactions. So types like A's, B like B, and there's the difference between them. And even this way, because these potentials are well trained, you can see that they remain the territories. They don't entangle together in that case. Like,
But
it's
now
important to say
sorry, I'm
here.
a bit confused. So the chromosomes, when I zoom in, they are built of the same things, the different chromosomes. So why do you have a force between those constituents turned on when they are inside the same chromosome, but not turned on when they belong to different chromosomes?
No, but the types in the interaction between chromosomes, they exist. The types is just universal, right? If
Right, the types is universal.
I am a type A, and I find another type A, I have an interaction between
different
them.
chromosome.
Even
Then
if
they
they are together or different. It's the same interaction.
But you tune those interactions between the types just looking inside one chromosome first,
Exactly.
right?
Yes.
Now that you know the interactions between the types by studying one chromosome, the first thing you showed was going to another chromosome and checking that now it works. And that was already nice, but okay. But now even more impressive is that now you don't care about about the separation between chromosomes, and automatically they decided to separate.
Yes.
That looks too good.
But
What am I missing?
it's not like we're not. You wait for something like that because you have this ideal chromosome interaction that's more related to compact the chromosome itself. So this helps it to not entangle together. This is one feature that you find, yeah.
So is there a force that maybe I
Make
thought
it separate is not.
no, the force only cares about the type, or does it care about the chromosome as well?
Just about the type. They don't, like this, type interaction that you call, they don't care about if they are chromosome 1 or 2. They just look for the different flavors and you have this interaction.
Great.
And
Then why do chromosomes 1 and 7 separate?
probably because the ideal chromosome potential. That is the
But
third
now
one.
you are saying there is a chromosome potential. It's not a type potential, it's a potential between chromosomes.
No. Let me go back in the potential explanation here.
So is the potential made out of terms between types, or is there also a term between chromosomes?
Like you have these three terms, right? The first one here is the type interaction. That's
Yes.
in the case of the multiple chromosomes. That is the case.
Yes.
No matter if they're chromosome 1 or chromosome 2, it's okay.
Yes.
This second one is just a loop term, so they just work inside just one chromosome. For each chromosome separately, you have this kind of loop formation. So we not have like looping being formed in different chromosomes.
So why? Why?
This is something that is not observable, observe in the experiments, for example. You not input that. If you look in the high C experiment, you see these loops, for example, is a classical very head dot in the high C. So it's just happened inside one chromosome. You not see this head dot between different chromosomes, right?
But how locally does the potential know if this green stuff is inside one chromosome or or if it's... It looks very non-local, this statement, that the force depends on whether you are inside the same chromosome or not.
No, like...
Am I missing? I'm probably missing because I don't know what is a chromosome and what's a gene, and I don't know anything, so...
No, like this kind of loop here, if you look the high C maps, what you can see, like in a very zoom-in region, you have this kind of formation here, right? Yes, this is the point. If I have like this kind of a feature that's happened in my experiment, I want you to observe that in my simulation as well.
He's asking a different question. He's asking, what's the physical reason why the loops only form inside one chromosome and they don't form between two chromosomes? He's asking the physical.
Okay, this is a very good question that you not have answer yet.
Okay.
right? This kind of mechanism is created. They just started to understand that called loop extrusion,
Okay, okay.
right? The mechanism how it's happened is until for now we will not know. I will show in the last part when I make a new kind of modeling that we start to observe in our results that is formed these stripes here. That's exactly the formation of these loops or not. The mechanism how it's work, what's the proteins that make this, I can get it for help. Let me get some, okay.
Then you are putting force that depends on the chromosomes, right? Your potential
Yes,
does
like
have
the last
forces.
term is just for inside the
Okay.
interchromosome
So
interaction.
that's why.
They not entangle, yes.
Okay.
So, like I explained, like
Sorry.
they have this proteins, this complex that called coenzyme, that they started to pull the protein and make these loops here, right? We will not understand. There's a lot of experimentalists, the Job Dagger, that the guy that helps to create the high C, they are put a lot of efforts to understand this mechanism, but we will not understand. You
Sorry.
want just to mimic what can happen that. This is the way that you are
A
modeling.
very trivial question. How can you form a loop between two chromosomes? A chromosome is a single line, right?
Yes.
And another chromosome is another line.
No,
So
no,
how
they
you
don't
form a
form.
loop
This is the
between-
question. This is
No, but even how can you think about a loop between?
It's like a piece of string, two different pieces of string that could be super separated. Why don't they feel an interaction? That wants to
Hmm.
Yeah, this is a way.
Exactly.
In that case, it's just a simple contact here. You just form and unform the loop.
Cioè, you need to close the loop, and for that you need a single chromosome, I think, right? Okay.
Okay. And this one?
So I'm just trying to understand how to get the dynamics. So you start with a 2D information,
I'm sorry.
then you go somehow to 3D.
Yes, in that case that you are working with the human chromosome. What happened?
How do you get this dynamics, the things moving? Are you solving some equations of motion?
No, this is like, okay, after you're training the potential in the microwave that I showed before, in that case, you just run Langevin dynamics with all the chromosomes together. And to create like this kind of high C maps here, you need to run like multiple replicas of the same chromosome, but in different initial states, to sampling as possible, like these contacts, mainly these inter-regions here between chromosomes is more hard. So it's intensive chromosome simulations. Yes, this is like a one of the runs that I run, right? So in that case, to form like to create like this map, you need to like more than 200 replicas to make sure that you find very well visit the landscape here. Yes. Yes, because what's happened is like this movement that you are something that you are studying now is like is the for example, if you get the blue one here, probably the chromosome one, because the size, they not move around all the directions here. Like you need to simulate for a long time to see it's happen. So to improve that this energy landscape, you make like more replicas with different initial conditions, just to get to sampling more these contact maps.
So you're solving the equation with how many?
Yeah, this is okay. What you were asking, okay, you started that, and then you make molecular dynamics, right? We solve Newton equations, and over the time for a period. What will happen instead of you solving it by kind of integrator like Leapfrog or another one that's more classical, you use Langevin because you not have like a solvent in this case, so you need to put it to use like a temperature regulator. You prefer use, in this case, Langevin dynamics. But in a sense, this is the process. You start that, you have the initial position, you select a velocity for each particle in some way. You can choose that by hand, you can randomize that, and then you start to make this, solve this Newton equation, change the positions over the time, right? And in some time, I get some frames of that. So I run for some time, take a frame, run for more long time, take another frame, and then I have this collection of frames, and then I can make my analysis, like create a high C, like for the structure, what's happened with the, for example, chromosome 1, go from one side for another, all this stuff. Is okay? Oh my God. So I will just show another, my computer, for some reason, is not working good. Okay, I will explain a little bit about a different work that is like this species here. This onager is a donkey that is very risky to be extinct. What's happened is they get some sample of this species in a museum and make the high C. Here is not good because I think the projector, but you can see that the high C map for that is very sparse. They don't have too much information here. And then, okay, let's take a look on that and try to modeling that to see what we can be obtained for that. So you make the same pipeline for the human one, where you get this first component of the PCA to get the sequence annotation of A's and B's. So you know what region here is A and B. Here is important to see because like all the information about the sequence annotation is coming from this region very close to the main diagonal, where I have the major information. You almost don't have much information here, like in the off diagonal. And what I can show here is that after you make the modeling using the same pipeline that I explained for the work before, you have this kind of map. If you can observe here that there are some regions that appears here in the simulation, but not in the experimental high C. And this raises a question. So this is like some artificial things that's happen because the way that I'm modeling, like the learning process of the high C is not good enough. So this, you observe this in different chromosomes. If you look in the chromosome 3, for example, here you can see that appear more regions that here I don't have. So this is a question that you raise. So maybe you are creating some artificial contacts that don't exist. And to make sure that it works, what's happened here, you make like a proof of concept, where I get, for example, the very good map, like the human one, and make a down sample of that. What means I start to remove information from this good map until reach, like in this case, 10% of the data, where this data here is look like the same that's happened in the onager map. Right? So, of course, we can remove this information in different ways here. But what I want to show for you is that when I get this map, just using 10% of the map and make the all pipeline for training, I have this as result, right? So this top part here. And then you can see, like, far from the main diagonal, you can see that you can see these patterns happen here that not appears in the 10% of the map. And then when you go for 100%, you can see that this kind of pattern that appears here, they appears in the 100% of the map. So now you start to believe that you can predict this region that is not well sampled in the experiment over this training, just looking for regions close to the main diagonal, you can recover some of that information. So when you go back to the onager, this region that appears in our simulation is look like they are real. They don't appear in the experimental maps because the lack of sample to do that. So it's a good way that you show that learning for regions here in the main diagonal can make us predict regions that is not well sampled in the experiment. This is a very good result that you have. Of course, talking about this open micron idea, you have a lot of another works that's running, like adding some different potentials in our.
Do you have examples where you did this type of predictions and then did it work?
Yeah, this is one of them, right? Because This is, you know the result. Oops.
Ah, so those were not from your simulations. Those were experiments. The results that you
The
showed.
10%, yes.
Sorry, the 10% was a simulation that you used to train.
Exactly. You
Then you
get like
trained-
a human high-C map here that have a lot of information and start to remove this information until this map just have 10% of the data. So this is the real high-C. This is the high-C that you are using for training.
Yeah.
Okay? And then you make the training. This is the comparison between the 10% of the experimental map and the result from the training, right? And here in the right side, I make the same. I training using the 100% of the data, and this is the prediction for the simulation. And what you can show that's training using just 10%, you have the same patterns that you find when you train in 100%. This is what you call prediction. So you can learn from regions that have more information that is close to the main diagonal. You learn for that, and then you can spread this information for long-range interactions. It's because, like in the modeling, you can sample much better than experiment that don't have this facility.
Have you done this with a blind experiment without knowing the 100%?
Yes. Okay. It can show to you that this is a first idea. When you do that, you make it for donkey. I just go a little bit further because you have, I don't think we can show all these results. But I wanted to show this result here. Okay. This is, I'm very happy because they just being accepted. Like yesterday, I received the email. But this is more bigger work because you make the same pipeline that I showed to Onegger. You made it for this woolly mammoth that is a 50,000 years extinct animal.
Sorry, why? Why are you choosing animals which are almost extinct or extinct? What's the reason for that?
I think the opposite question is better. Why not?
No,
right?
because
They
it's
find
much more-
these
no,
species
there is a
like
reason-
they are
sorry,
buried.
there's
Like
a reason why not? Because it's hard to reproduce, right? If the animal is almost extinct,
they are. Yeah, this one is extinct. Oh,
so you can't reproduce your experiment. Well, why did you choose something which you can't reproduce? What's the point?
I think that the best answer for that is like this kind of mammoth. They are extinct, but there are another species like another elephants that's derived for this mammoth.
I'm
right?
just asking, why not use the elephant? I'm just trying to
But
understand.
we are
what
using
the
the
reason-
elephant as well.
Okay.
In
never
that
mind.
case, in this paper that today, this week, they will be
Okay, but you told us
arrived.
about
You show exactly that.
Okay.
That this kind of species here, they remain a lot of information in the new kind of new species of elephants that they remain the same.
Okay.
And more than that, they show that like when you have this kind of animal that is frozen for 50,000 years, they remain that the DNA is not, the 3D structures of this DNA is well preserved.
No, I'm fascinated by extinct animals. I just don't know why it's related to your presentation. That's what I don't understand. You're trying to model chromosomes. I just don't know why you should look at extinct animals for modeling chromosomes. But okay, anyways.
Well, we are looking for a lot of different ways of the chromosome. This is one of them that appears for us that they start to create this that they call paleo high C. That is this is an animal that is very hard to find that, but this is just like a choice, right? You can do that for a lot of different species. Try to understand the concept of how it works. But here I just show you one example that we do. For example, you have like another experiment that is just started to running up, is that like now we are looking for like the yeast genome cell cycle. So they have this cell cycle that you need to understand what step is they can perform like high C synchronized in time. So you can see that there are different formations of region depends of the time of the cell cycle. What we are doing here is try to modeling that, to understand how the organization of the yeast chromosome is a multiple chromosome maps. Because here you can see that there are some features that's very interesting. For example, this region between chromosomes means that like the centromere of this chromosome interacts with the centromere with this chromosome. So what's happened in that case, like all the centromere regions for the different chromosomes like to stay together. And the regions of the telomere, that's the end of the chromosomes, like to stay in the cell wall here, right? So this is another example that you are exploring. Understand why they do this kind of structure formation. Okay? So I think I elongate. Yes.
It was not known before.
No, this is no for the experiment, for the images that the telomeres like to tether in the nucleus, right? And then we start to understand how it works. Because it's not like tether always. They are not tether stop, they are skip. They are moving, it depends of the time of the cell cycle, they change. They stop to stick in the cell nucleus and go inside the nucleus. So all these processes is not known for now. So you can see that we have a lot of questions that you not know their answer yet. You cannot go for the like, oh, I want you to modeling this way using first principles to understand exactly what's happened because you don't have idea how it works. We don't know how like this kind of chromosome interacts. So this is the idea that I try to pass for you that there are a lot of not answered questions that is important in all levels. You can go just an intrachromosome interaction. You will not have some answers. For example, the loop extrusion results. That is the last result that I will show to you today.
The point of that picture there, the things don't disentangle and they mix in the yeast.
They are more mixed than the human chromosome, and you do not know why yet. Like you make the same process for training the potential, the same things, but in that case, when you match the high C, exactly that's what happened in the experimental high C, they are more entangled. And then you're not, keeping, just beginning this work, so you not exactly know why it's happened. And why it's different for the human, for example.
Yeah, I'm still very surprised why the human separated by chromosomes, because it's true that there are some forces that only appear inside the chromosome, like you explained, but then, as Dario and Nathan pointed out, if they start locally creating a loop, it's not such a strong constraint that they are inside the same chromosome.
Yes, it's very weak, yes.
So why did chromosomes separate? Namely, let me maybe ask a different question. Suppose I don't paint by chromosome, but I start painting from half of first chromosome to half of second. I paint from half to half of each.
Okay.
How would the picture look like? Would it look the same, or would they look mixed?
They look more mixed, yeah.
They would look mixed.
Yeah, because like the half of the chromosome, they not separate in two parts, exactly the way.
So what physically, what distinguishes a chromosome? What is special about a chromosome compared to what is special from a region that's a chromosome from a region that is half of one chromosome plus half of the next one?
Okay, now you just go for imagination, the way that I think that it works, right? But like this kind of interaction, we are looking instead of look directly, we are looking in, how I can say that, in the implicit way. What makes one part of the chromosome likes to interact to another part of the chromosome is not a function of the chromosome itself. Probably it's the proteins that is around, mainly that interact with the histones, that push that to put it together or not.
So could you say it slower? Because they interact with what?
Like the interactions that make this chromosome moves, interact with different regions, is not an interaction made by the chromosome itself, but for the proteins that go around them. There's a lot, we need to remember that around the chromosome, there are a lot of proteins that interact with this chromosome and make this movement. So I believe that what we are looking in this kind of modeling, for this reason I call it a top-down interaction, is we have like implicit interactions. We will not know exactly what made this interaction, but we know that it's happened. This is the approach that José made in the protein folding theory for a long time. Right? I don't know why this amino acid interacts with another one. the way, when I say, I don't know how is this interaction. There are some tries, for example, the Leonard Jones that I explained here is a way to try to really understand what's the interaction. But you can create like a modeling that indirectly you know that these interactions happen, but you're not interested to know exactly what made this interaction, how is
So at some
the
point
first
in your
principle.
talk, you mentioned polymers. And I think from that point on, in my mind, I was just thinking of this picture. This is just a polymer.
It's just a polymer.
Well, but now you are saying, no, don't think of it just as a polymer. That's bad. Because if you think just as a polymer, you are never going to explain why region A and region B are separated. How should I then improve my picture? What's the minimal modification to the polymer picture that is required to start explaining why things factorize?
I think you need to include, for example, these proteins, right? If you wanted to make this interaction and understand exactly what's happened in terms of like first principles, you need to make a complete system, right? You have the histones, you have a lot of proteins that change this kind of interactions. This is another level of modeling. You are working in the kind of modeling that we indirect look this movement. Like, I'm not here to answer to you that, oh, this kind of loop extrusion happened because that proteins or that proteins in this way that is working. Like this protein push here, there's another way, protein push here. We're not interested on it. Of course, we are interested in that, but it's very hard to find these questions. What we are doing, like, oh, if I'm looking for the chromosomes, I can see that this kind of interactions happen very often because you see the experiment that's happened. So I'm creating a model that have an effective energy potential that reproduce this same idea. And then I start to understand what's happened. This case, for example, that I'm explaining about the loop here is something that I'm just starting to analyze. When you look for in vision, very zoom in this vision, you can see that what I'm drawing here, I have this drawn in this part here, right? And then I make some analysis over my simulations, and you can see, for example, that the formation of complete of this loop is just appear 14% of the time. It's very interesting because we are looking for some experiments that they measure if the loop is a complete form or not, and you reach the same results. So what I can say for that, I don't know exactly what we are forming my loop here, the loop formation here, but I have a modeling that can, in equilibrium, show that this happened in the same frequency that is the nature happened. This is just a first step to start to understand how it's happened. I don't know if I make it more clear for you, the approach of the way that you are modeling these chromosomes, right?
It's very impressive because you integrate out some degrees of freedom and you are left with an effective, purely polymer description, right? Where everything happens as an effective rule for a polymer.
Exactly.
But you are saying that there are effects that are as important as polymer physics, and yet you integrated them out, right? You remove them for effective things. I'm a bit unused to that. If I have, say, two particles of the same mass and I'm exploring energies where they are both relevant, if I integrate out one of them, I get a super messy description for the other one. I don't get a nice description. And here, you integrated out proteins that apparently have a huge effect, and yet...
No, but he had these extra terms. He had this
but it's funny that these extra terms, they are just local between the types.
No, they're not
where
just
this,
local.
no, it's not surprising?
I think it's more complicated. I think it's, I don't know, I don't follow all the extra terms, but I don't think it's just polymer physics. I think he has, well, I don't know.
It's not just polymer physics, because the potential was just a potential between different types.
The potential
and
that
for
I'm
forming
showing
the
to you
loops.
here
didn't
is
follow
like
all
the
the terms.
polymer interactions that like bonds, angles, and classical
it's
polymer
purely
physics.
purely polymer.
Okay,
Right?
and then you add on top of that these potentials that we get information using maximum entropy approach to learn that from the experiments that I see. For example, the micro one that I showed to you, you split this information that I extract from the IC in three different potentials: the types that measure like different flavors, the loops.
Okay.
That's very simple. It's just like
Yeah,
a
the
connect.
loops I understood.
And the ideal chromosome, that's one that is related to a genomic distance, right? This related to how compact are my chromosome. It's just that. This is my
so what's
full...
the third one? Sorry, maybe the third one is what I did not get.
Okay,
what's the third
I
one?
will show again to you. Just a second.
Is this third one the most important one for explaining why things separate?
Yes, I think yes. Like the ideal chromosome, that is this term that I'm using, they measure, like, for example, you have, if I'm looking for this, imagine that I'm looking for this bead interact with this yellow one here, right? The strength of these interactions is depend how far they are in genomic distance here. How beads they are separated, okay? And this
chromosome.
needs to be trained looking
an
for
interchromosome.
the experimental maps. So this
And
gamma,
D is big,
this is number of beads.
and it can be big.
Can
Just
be
look
big.
at the
They go
chain.
to
In your
the size of
experiment-
my, yes. But of course,
But
if-
only inside the same chromosome.
yes. But like this,
Then it's not polymers physics.
why not? Like this
Because a
potential, they have
polymer-
like these terms here. I show to you these potentials go this way. Like they have like a small interaction. It go to zero like after 10 beads. This is the way that this potential works.
But neighbors that are separate by 2 or 3 units only
They have,
in different chromosomes, it's 0.
yes, they don't have this. We will not put that. Let's make more clear now.
No, that makes clear. So then that effect is an effect that clearly that is not polymer physics, right? It's not a local term. It's not about extra information, about somehow you know that a bit of the polymer is in one chromosome and another neighboring bit is in another chromosome, and that you'll know about, right?
Yeah, but I really don't understand why it's not local, because it's just working like this small neighbor of the same chromosome. This potential is not work between chromosome.
Yeah.
No, yeah, I think it's clear. Like all these potentials here, if I remove the types here, is just between the same chromosome. You not have this interaction between chromosomes, just the types. That is a feature that having all chromosomes.
Yeah. So that was my question. Do the bits of the polymer, do they have a label that is just the type or the type and the chromosome? And
Type
the
and the
answer is
chromosome.
type and chromosome. But
Yes.
before you said just type. That was why I was very
Oh,
surprised.
I'm sorry. I'm
right?
sorry, baby. I'm not
Before
making
I
it
asked,
clear.
is the potential just between types or between
it's my
types and
mistake.
chromosomes?
I'm sorry
And
for
it's
that.
between types and
What
chromosomes.
I'm trying to say, just
Then it's
type.
not so surprising that things separate, because chromosome is one of the labels of the potential.
Right.
Yes, okay. I'm sorry, because what I'm trying to say, just like the types can be interacted between chromosomes. It's just that. The types can do that, but the other potentials is related to the single chromosomes.
Okay.
Okay. Okay. Okay. Okay, so now I just will finish, because I think we'll pass a lot of time. There's another works that maybe I try to include tomorrow when I try to explain a little bit about these potentials. But the idea is that like you create like a whole ecosystem to simulate chromosomes. Now you can train it. If you have some idea to include some potentials for our modeling, we can do that in a very easy way. We have a lot of collaborations with the experimentalists in the US. Here I have some of them. And like I said, one of my plans bring this kind of experiments to the Brazil. Like there's no theory group working this kind of modeling chromosomes here. It's something that is just start to bubbling outside, and I think it's a good idea to bring from here. And I think the IFT is a good place to start that kind of discussion. Yes, I think I not cover all the topics that I wanted. There are some details here, but I think you can give like idea so how this works. There are a lot of questions. This is the real. There are a lot of things that you not understand yet. You are starting for a very coarse-grained modeling to start to understand the basics and then give more steps, following the same way that we did with proteins, right? If you get the protein research, you have like 40 years of beginning of like very simple models, like and then nowadays you have like more very complex model to try to understand very specific things in proteins fields. Here's the same. We're just starting, just give the first steps. Okay, so if you have questions.
So maybe we can see if Hilda or Michael, do you have any questions?
It now appears.
A lot of questions, but I don't think I can formulate them in a very precise way. It's a very interesting subject.
Okay. Okay, thanks, Mike. Hilda, do you have any questions? Okay. Any questions?
No, I don't.
Okay, any questions from the audience? We had lots of questions during the talk. So we'll see him again tomorrow. Okay, so thanks very much, Antonio.
Thank you.